Hi everyone, and welcome to today's lecture where we will be continuing on with the theme of genetics. So now that you're all experts in the area of the processes such as transcription and translation, and that concept of gene expression, we're going to move forward and look at the various ways that bacteria and eukaryotes can control their gene expression. Because like we mentioned when we first introduced the topic of transcription, you don't want every gene in a body or cell to be constantly activated. Instead, you want some of them turned on at a certain moment and then turned off at another moment. So we'll go through the various ways that a cell can do that.
So now with these genetic processes, when you think about the processes occurring, there has to be some sort of regulation for them, keep them in check, right? So prokaryotic gene regulation is very important to the cells. Basically, what do they need at that moment? Do they need the products or do they not need them? And now with gene regulation, it can either be positive or negative. With positive control, think of it as the gene is only expressed if some sort of regulator stimulates it or activates it. Whereas in negative control, the gene will keep being expressed unless it's shut off by a regulator, meaning repressed by a regulator. So positive control, a gene needs to be activated, whereas negative control, a gene needs to be repressed. Now, in this process, keep in mind that prokaryotic genes tend to be organized in operons. And down here, we have a picture of an operon. It basically means multiple genes together under the control of one regulatory region. Now, with any of this kind of regulation, there are two terms you have to be able to distinguish. Some regulators can be cis-acting, whereas others can be trans-acting. What cis-acting means is that the regulator is actually a DNA sequence. So it's a DNA sequence on that chromosome involved in regulation. Whereas trans-acting regulations, when you hear trans, think traveling. Okay, trans are traveling because these aren't sequences. These are diffusible gene products. These are proteins. So trans-acting regulators are proteins that can swim around and bind to certain genes to regulate them. We'll see in a minute what examples are of these things. Now, like I mentioned a minute ago, when it comes to regulation, you could have things activated or repressed. And so the terms you tend to see is that a regulation pathway can either be inducible or repressible. A lot of times it's the end products that will determine what happens, but it varies pathway to pathway. But anyway, what's important here is to know that gene regulation can be positive or negative, so activating or repressing. That in bacteria, this gene regulation involves what's called operons, so a chain of genes. That the regulators can be cis or trans-acting, cis being DNA, trans being protein. And that it can be inducible or repressible, which you'll see in a minute. Basically, the expression can be activated or repressed. Now, to better understand this, we're going to go through the lac operon, and the lac operon that you see here, it codes for these genes here. You don't have to know the details about these particular genes, just know that all three are involved in breaking down and using lactose. That'll be important to help you figure out what's going on. So with the lac operon, before we go into the details of what everything is doing in this operon, it's easier to figure this stuff out if you first use logic. So here you have an operon that makes enzymes to break down lactose. So think about it. If there's no lactose present at that moment, why would you want to waste resources making enzymes to break down lactose when lactose isn't present to be broken down? Whereas if lactose is present, well, then you're going to want the enzymes to break it down and use that lactose, right? So that tells you that lactose is an inducer for this operon, okay? So we're going to go through the key players involved. When you look at this operon, you see a few different titles and names here. The first one is the repressor gene. So whenever you see the letter I in the lac operon, think of inhibitor. The I gene makes the repressor of this operon, of this whole regulation process. P gene stands for promoter, and you already know that a promoter is where RNA polymerase binds to express a gene. You then have the operator, so the O gene stands for the binding site where the repressor will bind, the repressor that was made by the I gene. Okay? You don't have to worry about L. We won't be going into that details at the moment. Okay? So focus on I, P, and O. So when you think of this lac operon, if there is no lactose present, which we see in this figure here, this shows no lactose present. As we said, if there's no lactose present, then you don't need these enzymes. It would be a waste to make enzymes that break down and use the lactose when there's no lactose there. So what happens in this process when no lactose is present, you'll notice that the repressor is bound to the operator. And when you have this big bulky protein bound over here, the RNA polymerase that binds at the promoter, it can't make it through this area to get to the genes to express them. So this operon now is repressed. There's no gene expression. Transcription cannot occur because a repressor molecule is currently blocking the RNA polymerase from making it to the genes. In this final picture here, which you notice now, instead lactose is present. And like we said, if lactose is present, well, yeah, now you're going to want to make the enzymes to break it down and use it. So when lactose is present, what you notice is the lactose will bind to the repressor molecule that was made by the I gene. And so when the lactose is bound to this repressor, it changes the shape of the repressor. We call that an allosteric change. So the lactose binding to the repressor changes the shape of the repressor. So what can't the repressor do now? It can't bind to the operator. And if the repressor can't bind over here, now the RNA polymerase is free to slide its way across all the way through all of the genes and transcribe them, which you see happening here. And ultimately, after transcription and translation, you get the enzymes that can break down and use lactose. Okay, so I recommend kind of pausing and rewinding and going over this slide one more time, just to make sure that you're very comfortable with all of the key players. And if you're having any trouble understanding this, just send me a remind message, and I'll try to make it a little clearer. To also help, I made an additional slide, which you'll find on the next slide, that kind of recaps what I just said, some of the key points. So as you can see here, I wrote down some of the key points, so you can pause the video and make sure you write down these notes on your slides. This is just a cute animation, which you can find on the Amoeba Sisters website, which has a lot of really good biology GIFs to visualize what's going on. And so this will help you visualize the lac operon and all of the key players. Okay, so here we have camp and cap regulation of the lac operon. Okay, this is going a little bit further in detail beyond the regular idea of the lac operon. This is basically to show you that even though a cell can use lactose and break it down and use it, cells actually prefer glucose, which makes sense because if you think about respiration, what do you always hear? You hear glucose. So if glucose is present, then a cell will prefer to use that and won't want to use lactose. And so what the figures here basically show you how what's called CAMP and CAP, which is glucose dependent, can help regulate the lac operon as well. And even though it's called a form of catabolic repression, you're going to see that it's actually an example of positive control because this is an activating or activator process. So like we said, glucose is preferred. And what was discovered is that polymerases don't actually bind efficiently to the lac operon unless what's called CAP is also present. So CAP stands for catabolite activating protein. And so the way you could think of it is, well, the operon basically thinks that if someone's wearing a cap, they're very attractive. Okay, wearing their little baseball cap, very cute. And so it will activate the lac operon. So if CAP is present, which we see binding over here, if CAP is present, then the polymerase works really well. It binds very efficiently, and you get very good transcription. If CAP is not bound to the lac operon right before it, then the polymerase will not be able to bind very well to the promoter, so you won't get a strong transcription because the RNA polymerase won't be that great at binding. So what happens in this process, if glucose is not present, in the absence of glucose, what's called CAMP complexes with CAP so that CAP can fit perfectly on the promoter. And when CAP is present, then the polymerase binds beautifully and you get transcription. If, however, glucose is present, that inhibits the enzyme that makes CAMP. And so that can't complex with CAP, and without CAP binding to the promoter region, there's nothing there to help the polymerase properly bind. And if the polymerase doesn't bind, then you don't get proper transcription. Okay, so I'm going to recap this on the next slide just like I did with the previous notes. Okay, so here you have a brief recap so you can pause the video and write down the notes in case you missed anything. Okay, so here we have another example of operon control and gene regulation. Here we have the TRP operon, or the tryptophan operon. Now notice this says tryptophan synthesis. So these enzymes are for making tryptophan. So again, you can use logic and figure, okay, if tryptophan is absent, well then we want to make it, the cell would want to make it, so transcription is going to need to be active. Whereas if tryptophan is present, well then, if you already have tryptophan, why would you want to make more? That would be wasteful of resources. So if tryptophan is present, then you're going to want this operon repressed. Okay, you don't want to waste resources making these genes or expressing these genes. Okay, so when you look at these processes, the option B here shows tryptophan absent. Like we said, if there's no tryptophan, the cell will need to make some. So you'll notice that the repressor is not the right shape in this scenario. It won't be able to bind to the operator, so the polymerase will get to bind to the promoter and express the genes. Whereas when tryptophan is present, what it does is it's a repressor. Okay, so the tryptophan will bind to the repressor. It will change its shape so that now it fits the operator. So tryptophan will allow the repressor to bind to the operator, and this blocks polymerase from getting from P through to the genes to express them. So transcription is blocked in this case. Okay, so I'm going to recap this in the next slide just to make sure that you have it in writing and that it's clear. Okay, so here you have the recap. Again, you can just pause the video and take down the notes just to make sure you didn't miss anything.
So now that you're experts with regard to the prokaryotes, such as bacteria, and how they're regulating their genes, now we're going to move on to eukaryotes, which includes your own cells and your own genes. Now, when we talk about eukaryotic gene regulation, it's first important to kind of remind yourself the idea of how DNA is actually packaged within eukaryotic cells. And we're going to look into the concept of transcription factors in a little bit more detail. So first of all, I want to point out that unlike with prokaryotes, eukaryotic genes are very difficult to access, meaning that it's very hard for the polymerase and the necessary molecules for gene expression to kind of get or reach the exact gene that you want activated. There are a couple of reasons for this difficulty of access. First of all, transcription factors, which are very important for activating and regulating eukaryotic genes, they're made in your cell's cytoplasm, okay? Whereas gene expression, when you're thinking of DNA and RNA, that's taking place within the nucleus. So these protein transcription factors that are made in the cytoplasm have to make their way into the nucleus to then reach the DNA in order to express the genes within that DNA. Secondly, eukaryotic genes are difficult to access because they are in nucleosome packaging. Okay, so I want you to circle star highlight the term nucleosome. What that means is that your genes or your DNA, as you can kind of see slightly in this picture here, your genes or DNA is tightly wrapped around various histone proteins in order to compact or fit all of that long DNA within your teeny tiny little nucleus. Okay, we're going to go over this in more detail in the upcoming slides, but first I want to go back and focus a bit on the transcription factors that we just mentioned. Now, like I said, transcription factors are very important for eukaryotic gene expression and gene regulation. Okay, there's various aspects of the transcription factors that must occur in order for them to really help gene expression occur or, kind of regulate whether it's activated or repressed. Now, first off, there has to be some sort of stimulus, okay? A stimulus will basically allow regulation in that it signals exactly which genes should be turned on and at what point in time. Now, like I already mentioned, transcription factors are made in the cytoplasm of the cell. So they need to be able to then translocate, meaning get into the nucleus. Once they're in the nucleus, they have to be able to recognize and bind to the DNA, as well as the transcription apparatus that we're going to go into more detail in a little bit. Now, when you look at this slide, I want you to make sure you remember that transcription factors, in order to properly regulate gene expression, need to be able to respond to a stimulus, or you can say respond to a signal, get into the nucleus, bind to the DNA, and interact or make contact with the transcription apparatus. Now, when I say respond to a stimulus signal, I want to point out that a lot of times this simply means the presence or an absence of a particular nutrient or substance. So for instance, when we talked about prokaryotic gene regulation, the idea of the presence or absence of things like glucose or lactose, okay, certain sugars, that can be the kind of signal that we are talking about when we highlight the idea of responding to a signal for transcription factors to actually, get to do what they do. Now, when we talk about transcription factors and their need to, bind to DNA and interact with the transcription machinery, I want to point out that transcription factors are what we call modular. And I want you to circle, star, highlight the term modular. What that means is the two domains, okay? So when you look in this picture, domains mean the two parts of a transcription factor protein do not need to directly bind to each other, okay? They just need the idea that the two domains have to be in close proximity to allow the factor to activate genes, okay? So basically, when you look at the figure on this slide, it's emphasizing that the domains are modular, meaning they are independent of each other. Okay, so for instance, the idea of GAL4, okay, you don't have to know, we're not in genetics or molecular yet, so you don't have to know these exact terms of GAL4, LEX-A, what they do. I just want you to notice that here you have a transcription factor with a GAL4 activator and a GAL4 DNA binding domain. Okay? Here you have what looks very similar, but instead of GAL4 and GAL4, it has one domain that's GAL4, one domain that's LEX-A. Now, because the domains are independent of each other, you do not need to have GAL4 matched with GAL4, okay? This second one is capable of activating transcription, but all that matters is that it's able to bind to DNA. So this one will only activate transcription when it finds a LEX-A DNA site to bind to, okay? Here, GAL4, the one with GAL4 and GAL4, will only bind to DNA when it has a GAL4 binding site, okay? But notice, either one, whether it's GAL4, GAL4, the matching domains, or the mismatched domains of GAL4 and LEX-A, either one can start transcription. You see that here in figure A and here in figure D, as long as they are able to bind to the DNA. So again, I want you to make sure to know that transcription factors have modular domains, which means that the domains are independent of each other. They just need to be in close proximity and allow that transcription factor to bind to the DNA. It's critical that they bind to the DNA. They do not have to match each other. Okay? Now, I also want to point out that just having these transcription factors is not enough to actually initiate transcription. Eukaryotic genes and eukaryotic cells also need what's called a mediator complex. Okay, I want you to circle, star, highlight the term mediator complex, and write the definition that the mediator complex is what recruits and stabilizes the polymerase at the promoter. Okay, and remember we said in the transcription lesson that the promoter is the part of the DNA that RNA polymerase binds to start transcription, okay? And every time you hear me say polymerase, remind yourself that's what's actually building the RNA, okay? That is what is responsible for the action of gene expression. So you need that polymerase to be able to get to that gene, get to that promoter. And the mediator complex allows for that by recruiting it and helping it stabilize, because it acts as a scaffolding that will hold the transcription factors close to the polymerase. Okay, so you see that here, the mediator complex here, you can kind of circle that to remind yourself this is the mediator complex, and it is basically a scaffolding or a bridge to bring the polymerase close to the transcription factors, okay? So it's kind of helping bridge everything together, okay? Kind of keeping things tight. Because remember, this whole process is about factors that are modular, meaning you want close proximity. You want a nice, tight complex of polymerase, your DNA, your gene that needs to be activated, and the various transcription factors and activators. You want them all close together, and the mediator complex helps do that. It helps pull together all of these factors. Just in case you had any difficulties writing any of that down, here is a slide where you can pause the video and kind of jot down some recap notes on what we just covered. Now, there are two other terms that I want you to be familiar with in gene regulation. The first one are the enhancers, which we've mentioned in previous lessons. So basically, enhancers, what they do is they bind transcription factors, and then they cause the DNA to loop so that they pull those transcription factors toward a promoter region, toward a gene to help activate that gene. And because they're looping the DNA to, bring these transcription factors closer to genes, they're able to help activate, one or more genes at the same time, any gene in the vicinity near them that they're looping to. Okay? Now, insulators are then responsible for helping to control those enhancers. So the enhancers are helping activation, and the insulators will then act as boundary elements, okay? They prevent enhancers from activating too many genes at once, basically from, they're preventing enhancers from activating too many genes that are further along the chromosome, okay? What they end up doing, so if you look at this figure here, enhancer sequences bind these enhancer binding proteins. And once they bind those proteins, that then causes a closed loop area, which means that the enhancer is not allowed to act outside of that loop or to reach any genes, let's say further along over here or further along over here. So thanks to these insulators and their insulator binding proteins, this enhancer can only help activate the genes within the loop, not further down along the rest of the chromosome, because you don't want the enhancer, activating all the genes along that chromosome. You only want it helping to activate certain ones. Okay? And insulators themselves can then be inactivated basically by methylating the sequences of the insulator. So in purple here, this would be the sequence of the insulator, that DNA. If you methylate it, then these insulator binding proteins can no longer bind, and it will no longer pinch together or keep that loop closed. Okay, so enhancers are inhibited or put under control by insulators, and then insulators are inhibited or put under control by methylation. Now, a lot of the regulation that we kept mentioning throughout these lessons have been kind of helping to activate and enhance gene expression. But there are also negative regulators, and the way negative regulators of gene expression work is that they tend to interfere with activators, meaning in some way they block the activator from being able to do its job of helping, initiate and enhance transcription. Now, the two ways that negative regulators tend to work is either by directly blocking the activator by taking up the recognition site on the DNA, or by messing or interfering with transcription factors. So the first method that you see here, so occupying activator recognition site, this is shown in this big figure here. okay? CTF, you don't have to memorize that name, but CTF is normally a transcription factor that binds upstream of a gene in sea urchins. And so this transcription factor is supposed to bind here to help activate transcription of the gene over here, okay? CDP, okay, again, you don't have to memorize the names of these letters, but this is to demonstrate negative regulation. Here, CDP is a negative regulator because it binds that same sequence that CTF is supposed to bind. And so now the transcription factor that should bind at this gene to help, activate this transcription, that spot is now blocked. It's like if someone parked in your parking space. So the transcription cannot bind to its proper DNA site because it's been already taken. Another way that a negative regulator can block a transcription factor from binding where it's supposed to is by forming a dimer, a mixed dimer, because remember, we said transcription factors have more than one domain, okay? And so what can happen is a negative regulator, like you see here in yellow, can bind as half of that dimer of domains of that transcription factor. And what do you notice about this negative regulator? It lacks that little green DNA binding spot. Without that DNA binding spot, this transcription factor cannot bind to DNA. Okay, so either the negative regulator will physically block a binding spot by taking the place where the transcription factor should be, or it messes up the transcription factor so that factor cannot bind even if it wanted to, even if the space is available. And we already said earlier in this lecture that transcription factors need to bind to DNA. Otherwise, they can't do anything. Okay? So that's how negative regulators tend to work. They block the transcription factors that would have helped or been required for activating gene expression. Now, when we talk about eukaryotic gene regulation, I want to remind you that eukaryotic genes, eukaryotic DNA, is in what we call chromatin structure. Okay, and chromatin is a repressive structure because, like I said earlier, chromatin or nucleosome structure, what that means is that your DNA, so the black lines in this figure, your DNA is wrapped tightly around, coiled around what are called histone proteins. And the reason why your DNA is all wrapped up tightly around these different proteins is to compact that DNA into a nice, small, compressed structure to fit within your teeny tiny nucleus of your cell, okay? But like I said earlier, the problem with this chromatin structure is that, look, the DNA is so busy interacting with those histone proteins, keeping it tight and balled up, that the DNA is not accessible to the transcription machinery we've been talking about, the transcription factors, the activators, the enhancers, and all of the different proteins and molecules that are responsible for activating transcription, especially polymerase. It can't reach it right now because the DNA is too busy hugging those histone proteins. So in order for each of your genes to be activated and expressed when we need them to be, chromatin structure has to be modified, meaning you need to loosen up this DNA away from the histone proteins so that it's nice and exposed, which you see here, so that the green bubbles represented here, those transcription factors can now come in and interact with the DNA. Okay, as opposed to when it's in the tight chromatin structure. In order to do that, your cells have two main categories, okay, to help manipulate the chromatin structure and loosen it up or move it either away or towards the histone proteins. There are chromatin remodelers and there are histone modifying enzymes, which we'll talk about in the upcoming slides. These histone modifying enzymes includes what we call HATs and HDACs. One acetylates, meaning it puts an acetyl group that you'll see in a minute, and one deacetylates. So these two here, HATs and HDACs, I want you to point out are opposites of each other. Okay? Those are the first ones we're going to talk about right now. HATs and HDACs, like I said, they're histone acetyltransferases and histone deacetylases, but we're just going to refer to them as HATs and HDACs. That's why I didn't write up their whole full name over here. Okay. What happens is that HATs, histone acetyltransferases, okay, so circle that one. HATs, they add what's called an acetyl group to the tail of the histone proteins, okay? Now the tails of the histone proteins have a positively charged lysine residue. And what do you know about DNA? DNA naturally has a very negative charge, okay? So that positive charge of the histone proteins under normal circumstances helps keep that DNA nice and tightly attached to the histone proteins because opposites attract. So the negative DNA is tightly attracted to that positive histone. When the histone acetyltransferase, the HAT, puts a hat on that positive charge in the form of an acetyl group, okay, we write it as AC over here, the hat covers up that positive charge of the histone by putting that acetyl group there. So now the negative DNA is not as attracted to that histone, okay? And so now it will loosen away from the histone proteins, as you saw in the previous slide, so that you have that exposed area now, okay? So you see it again on this slide. The DNA went from being tightly wound up in these coils very close together. Now the HAT, see HAT over here, the HAT added the acetyl group. So I want you to circle this part of the figure. The HAT added the acetyl group and now loosened the DNA away from the histone proteins. So now the transcription factors and polymerase can access the genes here and here to activate them. Now remember before I said that HDACs are the opposite of HATs. So as you can see from this figure, HDACs, the deacetylases, they remove that acetyl group, okay? When it removes the acetyl group, the positive charge is now exposed again. What happens when that positive charge is exposed? The negative DNA clings onto those histone proteins very tightly. And now, very tight clinging means that, again, the DNA is repressed because you can't get transcription factors or activators to fit in and reach it anymore because the DNA is being held occupied by the histone proteins. Okay? If any of this is unclear, please make sure to contact me and remind, and I will explain it a little further. Here is another recap slide so that you can pause the video, take some notes, since I know that some of the terms are a little crazy to spell. So this is to help you write down exactly what I was just saying. Now the other type of molecule or proteins that help to move around that chromatin structure are chromatin remodelers. Now when we talked about HATs and HDACs, those modifying enzymes were simply loosening the attraction or increasing the attraction of the DNA to the histone proteins. Chromatin remodelers, on the other hand, they're actually using ATP, they're breaking ATP to physically disrupt nucleosomes. And when I say nucleosomes, I mean the histones with the DNA wrapped around it. that tight chromatin structure. So rather than just loosening up the DNA, they're physically pulling that DNA off of the histone proteins. Okay, there are 3 models by which they can do this. There's the sliding looping method, where you see it basically tugs that DNA until the gene that you want to activate is now nice and away from the histone proteins. There's the ejection method, where the chromatin remodeler will pop out or pull away one of the big histone nucleosome structures. So you notice here you have 3 of those structures of histones wrapped with DNA. Here, the middle one has been completely removed, so that's ejection. And then you have repositioning or restructuring, where basically the chromatin remodeler will modify or swap out some of the histone proteins with variants, meaning other versions of histone proteins that do not attract the DNA or do not hold on to the DNA as well. Okay? So with chromatin remodelers, again, make sure you know that they are physically pulling apart or disrupting the nucleosomes, meaning the histones and the DNA, and that they use 3 methods, either sliding, ejection, or repositioning. So now that you've been introduced to a lot of the key players in terms of gene regulation and activating gene expression, I want to go through the steps that need to occur in eukaryotes in order to get transcription activated. Now, the first step is that transcription factors have to bind to the DNA. The reason that is keep in mind, eukaryotes don't have the sigma subunit of their polymerase, so they can't see the promoter without some help, and the transcription factors are their help to see and ultimately bind to the promoter. Okay? Now, in addition to the transcription factors binding DNA, the HAT, the histone acetyltransferases, then bind the transcription factors, and that brings them close to the DNA and the chromatin complex. And at this point, the HAT acetylates those histone tails, capping or putting a hat over the positive charge of the histone proteins. And now that loosens the DNA off of the histone protein. So now it helps expose the DNA so that ultimately the RNA polymerase can do what it's got to do. Now, around the same time, the chromatin remodelers are then recruited, and they help to physically disrupt the nucleosomes, to physically pull that DNA further off of the histone proteins. And so again, at this point, you're exposing more of the DNA, getting it away from those histone proteins so it's out there in the open, and so that now more transcription factors can bind. And by having more of those transcription factors there at that region, you are now allowing RNA polymerase to see the promoter because you have the help of all of those transcription factors. Okay, and the RNA polymerase is then able to bind to the promoter and kind of kick off transcription. But now keep in mind, we also mentioned that you need the mediator complex to help bring everything together, keep all of these factors and polymerase close together, close proximity, and to transmit the positive signal from those activating transcription factors to the polymerase to get things started. Now, with all of this, I want to point out that HATs, HDACs, the chromatin remodelers, they're not the only things that can modify nucleosome structure, that can work on those histones to affect how the DNA is attracted or repelled to those histone proteins. So we've already mentioned acetylation. So the hats help promote activation. So I want you to associate acetylation as a histone modification that promotes transcription activation, whereas deacetylase represses it. Deacetylase causes nucleosomes to be tighter. It causes DNA to be blocked from the polymerase. There's also methylation, and methylation can either help activate or repress transcription depending on which part is being methylated. So certain residues getting methylated will help activate transcription, others will help repress it. But in a minute, we're going to talk about how methylation is usually associated with gene silencing. Okay, so put a little star next to methylation and write that it is usually associated with gene silencing. So usually you hear it associated with genes being repressed. There's also phosphorylation, adding a phosphate group to the histones. And again, this one, like methylation, can either help activate transcription or repress it, depending on which residues of the histone, meaning which parts of the histone, get phosphorylated. There's also the term ubiquitination, okay, ubiquital group added on. Again, that's another example of activating or repressing. Now, we're not going to go into the mechanisms of each of these terms or what they truly mean. I just want you to currently associate them with histone modification and whether they activate, repress, or can do both. Okay, we go into the details of these in the upper level bio courses when you take genetics, molecular, and courses like that. The last ones I want you to see are the terms ribosylation, smolylation, and biotinylation. Okay, I just want you again to just associate these terms with modifying histones. Okay, we're not going to go into the mechanisms. In fact, for these three, the full mechanisms have not been fully elucidated by researchers yet. But again, I just want you to know that all of these terms on this slide are examples of ways that histones can be modified, meaning to modify those proteins of chromatin so that DNA is either tighter toward them and repressed or loosened away from them and now more active. Now, out of those terms on the previous slide, we've talked about acetylation and deacetylation a lot. The other one that I want us to go into a little bit more detail about is methylation. When it comes to methylation in your cells, there are two categories of methylases, okay? There are maintenance methylases, which means that the methyl group, which you see here, you can circle CH3, that is a methyl group. So you see this is methylation here and here, okay? Now, maintenance methylases, they add those methyl groups to new DNA strands opposite the methyl groups of parental strands. Meaning, if you remember DNA replication, remember it was semi-conservative? So those two parental strands separated, and then the new strands were built off of them. At the end of that DNA replication, you have hemimethylated DNA, meaning half methylated. The old parental strand is still methylated, but the new one hasn't been methylated yet. So maintenance methylases add the methyl groups to this new strand so that now, after DNA replication, after those maintenance methylases do their job, both strands of that DNA are fully methylated. The other types of methylases are de novo. And whenever you see de novo or novo, that means new, of new. So de novo methylases add methyl groups to completely unmethylated DNA. Okay, so when both strands have no methylation, that's when de novo methylases will do their job. So they're usually in, embryos and developmental parts or developmental stages of life. Okay, now when I pointed out these CH3 methyl groups, what did you notice? Each one of them is on the same type of sequence of DNA. We call those CG islands, okay? So CG islands are the target of cytosine methylation, okay? So what you see in that figure is that the cytosine, the C, in each of these CG islands is what gets methylated. And then from there, you end up having these be a recognition site for things like HDACs, okay? And so you end up having modifications of histone through recruitment from methylation. So methylation kind of ends up being important for recruiting other histone modifying proteins, and that's why methylation could then do things like gene silencing, because what do you notice? Well, that's an HDAC, and what did we say earlier? HATs help activate, HDACs end up repressing. Okay, so gene silencing means the repressing of genes. Okay, now when you look at this, I want you to note that housekeeping genes, the CG islands, are non-methylated. And what do you know about housekeeping or constitutive genes from our previous lesson? They are always active. So since we just said that methylation tends to be related to gene silencing and turning genes off, it makes sense that housekeeping genes are not methylated, okay? They're never methylated because that would silence them, that would turn them off, and you always want them on. So put a little star next to this fact and remind yourself housekeeping genes are not methylated. Now this slide I threw in to help recap the idea of maintenance methylases versus de novo. So the first bullet point here is to show that methylation is maintained in differentiated cells by maintenance methylases. So each round of DNA replication, at the end of that, when you have hemimethylated DNA, one strand, the parental, is methylated, the new strand is not, maintenance methylases will then get to work and make sure both strands end up fully methylated by focusing on the daughter strand that is not methylated. Then the second bullet point represents the de novo methylases, okay, in which I mentioned before the idea that during development, when you end up with double-stranded DNA that has no methylation on either strand, de novo methylases are responsible for taking care of that methylation. Okay, so maintenance methylases are only adding methyl groups to a single DNA strand. De novo are adding to both DNA strands. The last concept I want to go over with regard to methylation is an example of where methylation is extra visually important, and that is with the idea of X chromosome inactivation. So you notice male organisms, again, whenever we mention male versus female here, please note that we are talking about just chromosomes and the hormones that they lead to. You know, masculinizing or feminizing hormones. We are not talking about gender identification, what an organism identifies as. That's very different. But here, in terms of the sex chromosomes, an organism either has XY or XX. Now, in mammals, if you notice, if an organism has XX, when we looked at these chromosomes in a previous lecture, you notice the X chromosomes have a lot of genes on them, a lot more than the Y chromosome. So if both of these X's were active, then the females or any organism with XX would have a ton more genes being expressed than the males. That would be a problem, okay? So you need a mechanism to avoid the females or the XX organisms having double the amount of gene products from the X genes. So in some organisms, what they do is they cut the expression of the both X chromosomes in half and cut the expression in half of both X's so that they end up to equal one X, just like the XY organism. In other organisms, what they do is they double the X of an XY organism. So they make twice as much product to catch up to the XX. In mammals, what they do is called X inactivation. So one of the X's in any organism that has XX chromosomes, one of those X's is inactivated. The way that it's inactivated or completely turned off is through the methylation of what's called the exist gene, okay? The exist gene, what that does is it codes for a long, long strand of non-translated RNA, okay? So when the exist gene is active, okay, it produces a long non-translated RNA, and what that RNA does is it coats an X chromosome, okay? The other X chromosome, okay, in which exists is inactive, does not produce that RNA and does not get coded. Now, what do you notice in terms of if you're coding a chromosome? Well, you're blocking those DNA sequences from being accessible to things like transcription factors and polymerase. So this X that has been coded by exist is now inactivated, okay? Whereas the one that has not been coded by exist because exist is inactive, this chromosome is active. Okay, so exist and the chromosome, you can look at them as kind of being opposite. Okay, if exist is methylated, which you see in this figure here, this is the gene for making exist. It is methylated, so exist is silenced. Exist does not get produced. This X chromosome is safe and active, nice and accessible to transcription factors and polymerase. This second chromosome, when exist is not methylated, well, if it's not methylated, it's not silenced. It's very active. So this exist gene is producing a whole bunch of long exist RNA that doesn't code for anything. It's not producing anything, but it is covering and blocking that whole chromosome. This chromosome is inactive. Okay, so notice again, long exist RNA will coat a chromosome, making it inactive. Okay? It also recruits repressive silencing proteins to further inactivate that X chromosome. So I want you to note, kind of really look over this slide a few times, map out for yourself the idea of exist represses or blocks the X chromosome. So if exist is made and active, that X chromosome is inactive. It's blocked and repressed. If exist is methylated and silenced, the X chromosome is safe from that bad blocking exist. It is nice and active, okay? And all of this leads to this term, bar bodies. In any cell, the inactive X chromosome, they are called bar bodies. So when you hear that term, bar bodies, you can circle, star, highlight that. That simply means the inactive X chromosome, which has been coded by exist and it being blocked. Now, the reason why I said this is a very visual example is, for instance, if you have seen calico cats. Calico cats are always female. The reason that they are calico, meaning you see those two different colors appearing on them, is because their X inactivation can vary from cell to cell. So one cell, let's say over here, The X coding for orange fur with an orange fur allele might be active. The other one is coded by exists and turned off. Whereas in another cell, not far away from the orange fur cell, the opposite X can be the one that has been inactivated. So instead, in this cell, orange has been inactivated, and the X with a black fur allele is active. Okay, and so that's why you see that speckled pattern on some of these calico cats. Okay, if you have any other questions, feel free to contact me about this, but again, we're not going to go into the craziest of details in this course because a lot of this stuff you'll get to dive more into in genetics and molecular. Now, the last thing I want to briefly mention with regard to gene regulation is the idea of how cancer in general relates to gene regulation and defects in gene regulation. Because if you think about it, what do all cancers involve? Well, uncontrolled cell division, which is why you get a big mass of a tumor, because there's that uncontrolled growth. Now, the reason this occurs is that mutations, they can affect various types of genes, and the ones leading to cancer tend to affect the types of genes that either stop and slow the cell cycle, or genes that trigger cell growth and division through basically turning on or initiating specific parts of the cell cycle, okay? Meaning they either, these mutations will either block genes that are supposed to kind of stop cell growth, or they'll mutate genes encouraging extra cell growth. And either way, you end up with too much cell division and you get a mass or a tumor. Okay, now there are some terms that I want you to know. The first one is tumor suppressor. Okay, and a tumor suppressor would be any of the proteins that are normally, responsible for stopping or slowing the cell cycle, okay, which would stop or slow cell division, meaning cell growth, right? So that would normally suppress tumors, okay? There are also proto-oncogenes, which you can write as the first cancer genes, okay? These are genes that stimulate cell division because, again, keep in mind, cancer is uncontrolled cell division, okay? And then the last term that you see here, oncogenes, okay, that's once a mutation has converted that first cancer gene, that proto-oncogene, into an oncogene, which is the full-blown, mutant allele promoting cancer development, promoting this uncontrolled cell growth. Okay? Now, keep in mind, it's not just one mutation and boom, suddenly you have cancer. Ultimately, it's a whole bunch of mutations have to occur together to really encourage tumor growth, okay? So don't panic thinking, oh my God, all it takes one mutation and boom, I'm gonna have cancer. No, it takes a lot of different mutations that ultimately end up making conditions ideal for uncontrolled cell growth, okay? And again, whenever you hear onco, whether it's proto-oncogenes, oncogene, oncologist, onco is the term for cancer, okay? That relates to cancer. So oncologist studies cancer. Oncogene is the genes, the gene mutations or the mutated genes that promote cancer, okay? Here's just another one of those recap slides for you to pause and jot down some notes if I went a little too fast or if you had trouble with understanding the spelling of any of the words that I might have been saying in the previous slide. So now I want to give you a very common example of a gene that frequently is mutated, leading to, cancer, or, like I said before, since it's usually multiple mutations, this is just one of multiple genes that tends to be mutated in a lot of forms of cancer, and it is called P53. So I want you to circle, star, highlight P53 and make note that mutations in P53 tend to be linked with cancer. And the reason that is that under normal circumstances, so we all have P53 in our body, and normally it's induced or, activated when DNA damage occurs. And what P53 normally does is it binds to the enhancers of genes that are responsible for, stopping the cell cycle and for repairing DNA damage, and in critical situations where the damage is too bad, triggering apoptosis. Okay, so what that means is normally P53 is a protective protein for you. It's a protective gene product because if your DNA gets damaged in a cell, P53 will either help encourage it to get fixed or trigger apoptosis of that cell, okay, to stop the growth or trigger the bursting of that cell so that your body does not have problems with that damaged DNA, okay? Now, if P53 is mutated, what did we say in the previous slides about mutations in genes that are involved in the cell cycle? Well, if P53 is mutated and there's no active P53, now when DNA damage is induced, there's no longer this P53 product to stop the cell cycle or to burst and get rid of that damaged cell. So instead, if there's no longer a P53 there to stop the cell cycle, what happens? Uncontrollable cell growth, because you've lost that stop. Instead, the cell cycle keeps going and going and going and going, and boom, there is a tumor. Okay, so kind of jot down the notes on that, think about it for a second, and whenever you hear P53, I want you to think of what does it normally do? And then what happens when it gets mutated? Why does that lead to cancer? Okay, if you have any questions or uncertain about what I just said, feel free to contact me and remind. Okay, so this is just one more recap slide for you to pause the video and jot down some notes because, like I said, P53 is one of those terms that I want you to be able to quickly recognize. That is it for today, and as always, if you have any questions or you need a little help with some practice problems, feel free to contact me day or night in either the Remind app or through email. Thank you and have a great day.